Plasmonic biomedicine. Actually, the use of plasmonic nanoparticles for biomedical applications has been extensively researched, yielding significant advancements in the construction of ultra-sensitive viruses and the effective therapy. Actually, the unique surface plasma resonance phenomena of both plasmonic films and the nanoparticles with their exceptional absorption and the scattering abilities have much potential in revolutionizing diagnosis, treatment, and the evolution of diseases, in particular cancers. Here is the review of plasmonic virus sensor. Viruses, antigen protein, whole viruses, antibody peptides, complementary virus DNA, DNA peptide, nucleic acid PNA, virus-specific antibody, virus antigen protein whole virus, weakly bound viral infected cell, strongly bound healthy cell, complementary binding states, virus antigen protein whole virus, molecularly imprinted form. The plasmonic virus sensor. I can give you the overview of recent advancements of plasmonic nanoparticles in the fields of biases and the therapy is provided with an emphasis on the mechanism by which the plasmonic nanoparticles can be applied to enhance or provide signals for the detection of bioanalytes to treat diseases. Plasmonic biosensors work by functionalizing a surface with an antibody, which upon binding to an antigen results in variation in the angle of reflection, which represents changes in the resonance condition observed. What is plasmonic biosensors for food safety? This is a question for all of us. And the answer is also here. Plasmonic biosensors that can be applied to solid and liquid food analysis require few and the simple and maybe easily miniaturized. The aim of this review is to highlight the effectiveness of the plasmonic biosensors for detecting food pathogens present in contaminated samples. Food safety is becoming increasingly important to all of us, and because for food industry provide must quality products to minimize the health risks. Traditional methods to assure food safety, such as plate count and polymerase chain reaction, are accurate and robust, but can hardly satisfy the needs of the food industry because they are costly and time consuming. Plasmonic drug delivery system. Drug delivery systems offer efficient and localized drug transportation as well as reduce associated side effects in order to better understand disease. Precise observation of drug release and delivery is required. Here we present a strategy plasmonic to enable fluorescence imaging spectroscopy to track the release, the delivery of an anti-cancer drug doxorubicin from gold nanoparticles carrier in real time at a single living cell. A pH of responsive drug release profile was attained through the conjunction of doxorubicin to the nanoparticle surface via pH-sensitive hydrogen linkage. When DOX is bound to the surface of the gold nanoparticles, its surface enhanced Raman spectrum can be seen, but its fluorescence is quenched. So, the yielding significant advancements in the construction of ultra-sensitive viruses and the effective therapy. Framework of drug delivery system, you can see here, pump system, etc., injection loop, sample injector flow cell, and the angle scan, sensogram, in situ functionalization, space preparation strategies are here. Plasmonic photothermal therapy. Heat generated from plasmonic nanoparticles can be utilized in a photothermal therapy. A combination of near-infrared laser and the plasmonic nanoparticles is compelling for the treatment of brain cancer due to the efficient light to heat convenience and the compatibility. What is the principle of photothermal therapy? This is the photothermal therapy in which the cell use heat shock use photothermal therapy, and it is the 8.08 nanometer laser are present. Photothermal therapy is a cancer treatment that induces a cancer cell death by a heat generated in the tumor tissue exposed to near-infrared light. Near absorbents are used for facilitating the efficient heat production. Plasmonic photothermal nanoparticles. This is the plasmonic photothermal nanoparticles. Plasmonic excitation T is equals to 0 second. Landau damping T is equals to 1 to 100 frequency. Carrier relaxation T is equals to 100 to 1. Thermal dissipation, population, and the population for all of this is here. You can see this easily. The PPT means photothermal therapy, plasmonic photothermal. Cancer diagnosed and into the next time targeting nanogold particles and move to the next step moving on the tumor and the next step is like the exposure and the next step is recovery. Photothermal therapies for cancer. Actually, the rapid relaxation of these excited electrons produces strong localized heat capable of destroying strongly targeted cancer cells via hyperthermia or other thermal-based effects. Is thermal induced by the plasmonic gold nanoparticles. We start plasmonic for a thermal therapy. This is the and move towards the next section. So this is the plasmonic hybrid systems. You can see here, this is the plasmonic hybrid system. This is our next topic. We can discuss here. Plasmonic material with nanomaterials. The fundamental basis of such work is to link unique phenomena that arise at the nanoscale to the presence of a specific analyte biomedical and to modulate the intensity of such phenomena in relation fashion. In direct proportion with analyte concentration, precise engineering of nanomaterials is out of the most important here. So this is the plasmonic polymers. Plasmonic polymers are quasi one-dimensional assemblies of nanoparticles whose optical responses are governed by the near-field coupling of localized surface plasma. So this is the structure of the plasmonic polymers. Actually, the plasmonic refers to a system that combines plasmonic materials or structures with other components or functionalities to create new or enhanced functionalities. Plasmonics is a field of study that deals with the interaction between light and metal and nanostructures known as plasmons. As we know very well about that, plasmons are collective oscillations of electrons in metals that can confine and manipulate light at the nanoscale. Plasmonic quantum drives. Plasmonic devices confine light to nanometer-sized regions of space, which turns them into effective vector cavities for quantum emitters. Basically, a plasmonic system typically involves integrating plasmonic materials or structures with other materials such as semiconductors, dielectrics, biological materials to achieve specific objectives. One example of a plasmonic hybrid system is the combination of plasmonic nanoparticles with semiconductors materials in the field of solar cells. What are the 2D plasmonic materials? Plasmonic materials, graphene, oxides, antigens, and the HBN are just a few of the commonly utilized plasmonic materials described in this section. This is the 2D material structure. The two-dimensional materials are defined as crystalline materials consisting of single interatomic interactions are much stronger than the stacking direction. A hybrid pairing inorganic 2D materials. This is all hybrid pairing materials and these materials are inorganic and the two-dimensional materials. You can see here. And this is the blue one, this is the red one, this is the yellow one, and these are called the multi-shades hybrid pairing structures. Now, this is the last and the important one section of this topic, latest research on plasmons. Actually, the developments in the study of plasmons, which was very needed to be known that it has been known for many years that metallic nanoparticles can utilize various chemical reactions both in the dark and the under medium possibilities. Possibilities for application in the various fields are here. These clients were challenged in a recent series of papers. Specific experimental setups to discover them in chemical reactions were examined in detail. The conclusion that arose from these works is that as long as temperature gradients exist inside the system, a quantification of non-thermal effects close to impossible. Instead, a standard thermal theory was shown to be capable of explaining the experimental findings quite accurately. Here we review the central lines of thought that led to these conclusions from a personal point of view. We lay out the key aspects of the theory and point to the specific caveats one must be aware of in performing photocatalysis experiments. Finally, we provide some future directions of study. Here is the plasmonic structures. You can see here source, simulation region, rectangular box, copper structures, substrate, and now this is the monitor. These were the nanoparticles and these were the nanoholes array, and it is abbreviated as NHA. Plasmonic resonance energy transfer. This is the energy transfer diagram. You can see here. This is the acceptor graph and this is the donor graph. This is the absorption graph and this is the acceptor or the donor graph both. Plasma resonance energy transfer. Basically, plasma resonance energy transfer is the energy stored in the collective movement of free electrons in metallic nanoparticles being transferred to the absorbed chemical environment with matched electron transfer. Plasmonic quantum optics. Quantum optics is a rapidly growing field of research that involves the study of the quantum properties of light and its interaction with matter at the nanoscale. So this is all from my side. This is my presentation topic, which we are discussing about the plasmons and the research of plasmons. And these are material that are conducted from these references, books and overviews, and then articles from these books and these encyclopedias in partial chemistry, methods in thermology, encyclopedia of modern optics, comprehensive and analytical chemistry, and these were books that were published by the scientists. Thank you so much. Thank you so much, guys, for listening. Allah Hafiz.